Welcome everybody, I hope you're staying safe, doing well at home, coping with all the tedium of being at home, but hopefully you've got plenty to do. This is also going to be the first of hopefully several biology year 12 revision lessons that I'm going to do. I'm going to start in this lesson looking at biological molecules, and we're going to look at our three main groups, our carbohydrates, lipids and our proteins. And I'm going to start of course with lipids, which I think in some ways are slightly easier. So our lipids are quite simple in many regards. They're not polymers like many of our other biological molecules. They're made up of smaller subunits. So the two main types of lipids that we're going to look at today are our phospholipids and our triglycerides. Now both contain a glycerol group, which I'm going to draw very simply as this sort of blob here, but you can appreciate the structure is more complex than that. So I'll draw two of those, one representing the phospholipids and then one representing our triglycerides. So both have a glycerol group. Okay. The phospholipids, in addition to that, will have two fatty acid chains bonded to the glycerol group. And of course the bonding that we would see here is an ester bond, which would form as a condensation reaction once the fatty acid bonds onto the glycerol. In addition to that, of course, we have this phosphate group as well. Okay, which gives the name of the phospholipid. So to summarise, with our phospholipid, we've got a phosphate group, a glycerol molecule, and two fatty acid chains. So that is a phospholipid in terms of its structure. Triglycerides, again, have that glycerol. They have no phosphate groups. That's one key difference between the two of them. Another really key difference you can see here is that they actually have three fatty acid chains, hence their name triglyceride. Okay, so that's our structure of our triglycerides. So often in the exam you're asked about the key similarities and differences. Key similarities, of course, they both contain glycerol, they both contain fatty acids. The fatty acids are bonded onto the glycerol via condensation reactions and form an ester bond, probably more likely to get the mark for the ester bond there. But where they start to diverge in their differences structurally is in the phospholipids. It has a phosphate group, which of course a triglyceride doesn't have. Always make your statements as comparative as possible. The phospholipids only have two fatty acid chains, whereas the triglycerides have three. So that's an overview of the similarities and differences in structure. In terms of the roles they play, their sort of functional roles, phospholipids are polar in nature. In other words, they have this hydrophilic head and these hydrophobic tails. The hydrophilic head is attracted to water molecules, has an affinity for them, so it's attracted, whereas the tails are hydrophobic. They're repelled by water, they want to get away from water. So what they do is something quite unusual. They will group together and they actually form what we call a phospholipid bilayer. So what you would see, if I draw a very simple diagram of these, is that these heads would be sticking out towards the water or towards the cytosol, the cytoplasm, like that. And of course they will form a secondary layer like this. And this ensures that the heads are always, get rid of that triglyceride, the heads are always in contact with water and those hydrophobic tails are away from it. And that is a self-assembling structure, quite remarkable, a bit like how a soap bubble forms. You know, if you're blowing bubbles as a little kid, you still might do it now for fun, you know, I've been known to. They'll actually self-assemble in that simple way. Okay, so that's one of the primary roles there of our phospholipids. They will form a phospholipid bilayer, which will help form cell surface membranes or membranes around organelles as well. Triglycerides, their function is quite simple really. They are generally considered to be an energy storage molecule. So in your adipose tissue, which I've got a fair bit, especially with the current limitations on exercise, I built quite a bit up there. The triglycerides, I'll have quite a few stored in those cells there, and they're also used as an energy storage molecule. And they get broken down, the glycerol and the fatty acid get separated, and then those fatty acid chains can then be hydrolyzed to release energy during aerobic respiration. I'm not going to go into epic detail about the oxidation of those fatty acids here. That's more of an A2 topic and something we will investigate later in the year. Okay, so that's a nice brief overview of the two main types of lipids, but bear in mind there are a few others too, such as cholesterol, which helps to maintain or reduce the fluidity of cell surface membranes, and that will fit quite nicely between those phospholipids, making them slightly less fluid. And of course we have various other steroid hormones like testosterone and estrogen, which are also lipid based and are lipid soluble. So we'll move on now from our lipids and we'll have a look at the slightly more challenging carbohydrates. Cheeky bit of tea break here. King of the melts. All better. So I'm ready now for our next group of biological molecules, which are our carbohydrates. Okay then, so carbohydrates, or polysaccharides, as they're often, polysaccharides, disaccharides and monosaccharides, as their collective terms are very important molecules. I'll start with the most basic, which are the monosaccharides. There are three basic types of monosaccharide you need to be aware of. That is glucose, fructose and galactose. So monosaccharides are generally soluble, they're single, what's the best word? Ringed molecules, I would say, is probably the best way of doing it. And obviously they provide an energy source within our cells, and of course they are the primary respiratory substrates within our cells. I'm now going to draw how our disaccharides would actually form a bond. So I'll start off by drawing some very simple monosaccharide, like this, which is a hexagonal shape, sometimes called a hexo sugar. And this one's going to be alpha glucose. And what we will see here is that hydroxyl group is below the ring, and another good way to remember that is ABA, which I'll put over here. So ABA, alpha below, beta above. Okay, it's easy to remember that one. So then we'd have another adjacent alpha glucose like this. Well, that's a very terrible hexagon. I'll redraw that one to go back and do some geometry, I reckon. Okay, there we go, that's a bit better. Like that. And again, over here we'll have that hydroxyl group below the ring there as well. These two hydroxyl groups here can help form a glycosidic bond. I can remember it there myself. Glycosidic bond through a condensation reaction. So often what you have to do in the exam is actually circle where that would occur. And in a condensation reaction, a water molecule is removed. So remember, water has the formula of H2O. So I'm going to redraw that there so it's a bit more accurate. Don't worry, that will already be there for you in the exam, generally speaking. And I will circle what will be the water molecule that's removed. What will remain will be something like this. And we'll have our oxygen there still intact, and obviously the removal of that water. So that is a condensation reaction forming a glycosidic bond, more specifically a 1,4 glycosidic bond, because it's carbon 1 carbon 4. So here we can see I've formed a new disaccharide with 2 alpha glucose, which would be in this case maltose. The other 2 disaccharides that you need to know are sucrose and lactose, which is found in milk. Okay, so that's the basic overview of our monosaccharides and our disaccharides. Polysaccharides, only rule 3 that you need to be aware of, and they are starch, cellulose and glycogen. We'll start off with starch. So starch is a polysaccharide. It's made up entirely of alpha glucose molecules. And alpha glucose, because both of the hydroxyl groups are below the ring, it forms nice easy bonds. There's no flipping or anything required. But interestingly, even though they kind of all line up, it actually causes the molecule to actually coil. So starch will have a very kind of coiled appearance like this. Okay, so in terms of that's what its structure looks like. There's some hydrogen bonding in there holding it together. Now, that structural arrangement is very important because it actually gives starch a lot of its unique properties. So starch's main function in plant cells is to provide an energy store for that plant. Having this shape really makes this molecule ideal. It's coiled, which makes it very compact. It's insoluble, so therefore it won't affect the osmotic potential of the cell. And it has got some branches on it occasionally, so amylopectin and so forth like this. And that will obviously increase the surface area for hydrolytic enzymes to break those bonds by hydrolysis. So then it can provide a very rapid source of glucose for respiration within the cell. So that's starch. Okay, Our next polysaccharide we're going to look at is cellulose. Now, cellulose is actually formed from glucose as well, but it's made from beta-glucose molecules. Now, with beta, and if I go back to my ABBA like I did before, always very helpful this, so beta above, you'll see that actually this time, I'm going to draw the endpoints on my beta-glucose, that the hydroxyl group is above there. And over here, this one is actually below. So you can see actually this time we can't form any condensation reactions, we can't form any glycosidic bonds at all. So what has to happen is one of these molecules has to flip a full 180 degrees in order for that bond to form. So if I draw that in, you'll see, you'll get the idea. So once that's flipped, our hydroxyl group then fits up. We can form that glycosidic bond, bang, and we can form a chain. Now, through this alternating bonding going on in cellulose, it actually results in these very long, straight chains. And remember, describe cellulose as long, straight chains formed from beta-glucose. So again, I'm not going to draw them in epic detail, but imagine they look like that, very straight linear chains. Now, on their own, they're relatively strong, but it's when they work together. So what you'll actually find is multiple chains will actually be bonded together by hydrogen bonds, or cross bridges as they're called. So hydrogen bonds on their own are pretty weak, but when you've got many of them, thousands, millions of them, they actually become very strong. And when you start getting a lot of these chains together, you form what we call microfibrils, which are much stronger, and they will actually form a mesh-like network within the cell wall, which makes it incredibly strong and durable and helps give that structural support for the plant cell wall. So that's our cellulose. One last thing to say about cellulose is we are completely unable to digest cellulose. We can digest starch, fine, we've got the enzymes for that. We've got amylase followed by maltase to break it down. But we cannot break down cellulose. We lack the enzymes to do that. We don't have, none of our enzymes have the correct active site, which is complementary to any of this in here. Even though this is made entirely of sugar, it's completely inaccessible to us. And of course, cellulose forms the fibre within our diet. So we'll move on to look at our last polysaccharide quite briefly, and that is glycogen. So glycogen, I always think, is the kind of animal equivalent of starch. So it's that energy storage molecule within our cells, particularly in our muscles and our liver cells. They've got large stores of glycogen, so they can obviously provide glucose when required for respiration. And in some respects, it's similar in terms of its properties to that starch. It's very compact, means it takes up far less room in the cell. It's insoluble, so it won't affect the osmotic potential. And it's branched. Now, it's far more branched than cellulose is. So you would see very common. I'm drawing a very quick diagram here, but it's got multiple branches, again increasing that surface area significantly for enzymes to begin breaking this structure down to release large quantities of glucose. So that's what glycogen is in terms of its structure, very similar to starch in many ways, and of course its function. The only real big difference between starch and glycogen is it's highly more branched compared to starch, which has some branches but not to this extent. So that deals with all of our carbohydrates in detail. We'll move on to next, which is our proteins. So we're now going to move on to our last major group of biological molecules for the proteins. But before I start that, a cheeky cup of tea I think is in order. Thank you, Mrs. P. Very kind. Down in one. That's how hard I am. Right, okay, next one then is proteins. Okay, so proteins are polymers. They're made up of many monomers bonded together, and the monomers that form proteins, of course, are amino acids. Now, amino acids, you need to know their general structure, and you have to be able to draw it as well. So you start off over here with nitrogen, and what I do is do the backbone, and I fit in the other bits as well. So you've got this NCC backbone to it like that. At this end here, you'll have two hydrogen bonded onto the nitrogen like that, and this bit here is called the amine group, or the amino group if you like. We then have our central bit here, which is called the R group or the variable group, because this R here represents a wide range of different structures depending on the type of amino acid in question. And we have our last bit here, which is called our acid group, okay, which then gives you the name of amino acid. So all proteins in nature, and there is probably an unlimited variety of proteins that you could construct, are all made from these types of amino acids, and there's about roughly 20 different types. And depending on the sequence you put them in will determine the overall properties of the protein that we can form. So I'm going to start off there now looking at the polypeptides, the proteins, and talk about their 4 main levels of structure. So we'll start off what I call the primary structure here. Now the primary structure is very, very important you get this right, because it has huge ramifications in many, many topics. So you need to be able to define this precisely. So my simplest definition for that one, for primary structure, is the specific sequence of amino acids. So that is determined, that specific sequence of amino acids is determined by the DNA base sequence. The sequence of those DNA base nucleotides will determine what sequence those amino acids will go. Now once you form this long chain of amino acids in ribosomes, the actual molecule will then begin to start initially folding, and this will form what we call the secondary structure. So the secondary structure is the initial folding or coiling of the molecule held together in place by hydrogen bonds. Now the 2 sort of primary types of secondary structure you'll see are alpha helix or beta pleated sheets. Okay, so I'm going to define that now. The initial folding or coiling held together by hydrogen bonds. So now we'll look at the other 2 levels of structure, and that is our tertiary structure. Again, very important, this one. And this is where we get further folding of the molecule. We end up with that 3-dimensional shape, and it will be held together by 3 types of bonds: hydrogen bonds, disulfide bonds, and ionic bonds. Now, where do all these bonds come from? Why are they happening? Well, it all comes down to that R group I referred to in the amino acids. So each amino acid will have its each type of amino acid has its own specific R group, and some of them, for example, like cysteine, will have sulfur, and that can then form a nice disulfide bridge or bond across the molecule. And if 2 of those are either side of the chain, so you imagine my hands, imagine this is one here and one over here, and what will happen is they're attracted to each other and they form a very strong disulfide bond. And of course that's folded the molecule around. Rather than being straight, it's now looped like this, and this is obviously happening multiple times. So this is our further folding. We end up with our 3D shape, and it's held together by, and I would always name 1 or 2 of these: hydrogen bonds, disulfide bonds, and ionic bonds. Okay, and in a moment I'll just draw you a very simple diagram of the tertiary structure. So when I think of tertiary structure, immediately I start thinking about enzymes, which are of course a very important group of proteins. Not the entire reason for proteins. Proteins have multiple roles. So they're hormones, they can be antibodies, they can be for binding and receptor sites on the surface of cells, just to name a few. They can be structural, like collagen. But enzymes is one of the main areas that we tend to look at. So if I was to draw an enzyme, and we'll look at enzymes in more detail later in the video, you would see that I would have a polypeptide strand that's going to be folded something like this. So it looks a bit of a mess really based on that. But remember, this would be a 3-dimensional object. And within that, to get this kind of folding and stuff like that, we would have various types of bonds. We'd have hydrogen bonds in there across the chain, looping them together. We'll have some disulfide bridges. We'll have various sort of ionic and ionic bonds in there as well, holding the whole thing together. And obviously here, this would represent possibly my active site where my substrate would bind with that enzyme. So that gives you a nice kind of overview of how the bonds create that final 3D shape in the tertiary structure. So the last one to look at, of course, it's in many ways the simplest, is the quaternary structure. Not all proteins have a quaternary structure. Some do. Hemoglobin is a good example of this. And the basic definition for the quaternary structure is where you've got 2 or more polypeptide chains bonded together. So imagine that being one polypeptide chain. You just have multiple of these bonded together to form a more complex structure. So I'll write that definition down. So 2 or more polypeptide chains bonded together. And I'll just do a very quick drawing of something like hemoglobin. Forgive me if I've got this not entirely correct. So in the case of hemoglobin, we've got 4 polypeptides bonded together, 2 alpha subunits, 2 beta subunits, and don't get these confused with alpha helix and beta pleated sheets. These are very different structures. They are beta pleated sheets and alpha helix are secondary. These are entire polypeptides in their own right. They may well have alpha helix and beta pleated sheets within their structure as well, but we just call them alpha and beta subunits, which can be a little bit confusing at times. So that's a nice overview of proteins. We've also I briefly mentioned some of their specific roles. I'll just recap that. So they can be biological catalysts, i.e. enzymes. They can be structural, so they're providing support, like collagen. They can be used as antibodies, which can help destroy pathogens like viruses and bacteria. And of course, they can also be hormones as well, and receptor proteins, binding sites for other molecules as well. So they play a very, very critical role within cells. They're really kind of the machinery that makes cells function. So the last little bit of this video I want to talk about is probably the more complex aspect of it, and that is enzyme structure. So a little bit more about specialized proteins. So we'll have a look at that in a second. One key thing I forgot to mention is about how polypeptides themselves are bonded together. How do we form that primary structure? And that again is through condensation reactions between adjacent amino acids. And the bond that's formed from that condensation reaction is called a peptide bond. So don't forget that. So a quick review of the 3 types of bonding we've seen so far in this lesson. So in lipids we had ester bonds. In carbohydrates we had glycosidic bonds. And in our proteins, we have our peptide bonds. So let's look at these very interesting and very, very important molecules of enzymes. Now, I think for many people, they think about enzymes and they just think, oh, they help digest your food and that's it really, that sort of basic thing, or they don't really fully understand them. But I cannot emphasize enough how essential enzymes are to biology, to life. It would not be possible. Cells would not even be able to function on that kind of cellular level if it wasn't for these amazing kind of nano machines that are enzymes. They are really, really essential to life. They can help build up molecules, they can help break down molecules, they can change molecules from one type into another, and they speed up chemical reactions up to 14 million times faster than they would otherwise occur. One of my favourite sort of things I calculated once is, let's say it takes 8 hours to digest a meal. Without enzymes to help you digest that food, it would take over 2,000 years to digest it. Now, I don't think you'd be getting sufficient energy out of your food if it took you that long. You'd be long dead before that even happens. So enzymes, play really important roles. They help with DNA replication, of course, breaking down food molecules, building cells, maintaining chemical reactions, everything that is going on, anything that needs a little bit of order and a little bit of structure and speed to the process in terms of the chemistry is all about our enzymes. So at GCSE, you should have learned about the lock and key model, and it's nice and simple, nice little idea really. In reality, you have our enzyme, looks like Mr. Pac-Man here. They look nothing like that at all. Hopefully you've got an idea now that they are actually kind of these polypeptide chain folded up, and the key bit is the mouth on Mr. Pac-Man, and that's the active site. And the idea with the lock and key model is very simple, that the substrate has a complementary shape to the active site. They fit together perfectly, and then by some process that would break down that substrate. Well, actually, before that would happen, that substrate would collide with the active site, forming my favourite term, enzyme substrate complex. I'm going to write that ESC on there. Please write enzyme substrate complex in the exam. Don't write that. The examiner's not necessarily going to know what it means. You and I do, but make sure you write it properly. So that enzyme substrate complex would form, and then it would break the substrate down and would end up with our products from that reaction. Okay. So that's a very simple lock and key model. Let's fix Mr. Enzyme. Like that. Okay, all right, for GCSE. Unfortunately, it doesn't explain one of the most important aspects of enzymes, and that is how they lower the activation energy in a reaction, because they are catalysts. All catalysts lower that activation energy down. That's how they're able to get a kind of able to react and catalyze these reactions at much lower temperatures and normal pressures. So this model doesn't really explain that. How would it sort of fit together and break the bond? Doesn't really explain it. So instead, we look to what we call the induced fit model, which makes a lot more sense. And the idea this time is that my enzyme will have an active site that is, somewhat complementary to the substrate. So I'll try and keep things the same as before. This time you can see that the substrate is not going to easily fit in there. But upon collision, the enzyme's active site will change shape to accommodate that enzyme. Now it doesn't, that substrate, sorry. Now it doesn't necessarily want to do that. It's under sort of duress, if you like. And that will actually apply force to the substrate molecule like this. I almost think a bit like if I looked at my hand like this. Imagine it's sort of this shape, and here's my substrate, this meaty fist here, goes in, and actually will actually cause it to change shape and sort of mould around it like that. So like that. And now that's putting pressure, that's applying force onto that substrate, which is therefore lowering that activation energy down to make the reaction happen. So that's our induced fit model, which then therefore explains how enzymes are able to lower the activation energy. So what else do we need to know about our enzymes? Well, quite a lot really, and they come up all the time, but obviously I'm not going to be able to spend 2 hours talking to you about them, so we've got to keep things brief. So first of all, we've got to think about which factors will affect how our enzymes function. So the first, most obvious one, hopefully one from GCSE you're familiar with, is temperature. We've also got to think about pH. We've also got to think about substrate concentration and the enzyme concentration. So I'll start with temperature. So temperature is pretty straightforward. Initially what you would see, I have a little graph with that, you would see that as I increase the temperature, there's temperature on the x-axis, you would see that the rate of reaction with the enzymes would increase, reach a sort of point, kind of a peak, and then would start to rapidly drop off. So the increasing region, what's happening there? Well, as you're increasing the temperature, you're increasing the kinetic energy of your particles, our enzyme and our substrate, like this. So as we're getting warmer and warmer, these things are moving around more, so therefore they're more likely to collide and form more of those lovely enzyme substrate complexes. So if you imagine at low temperatures, reaction, as I start to warm things up, the reaction gets faster and faster and faster. But why doesn't this carry on? Why don't we get this nice kind of exponential increase forever? Well, hopefully you're well aware, the enzymes will begin to denature. And what's happening there with denaturation is that increased kinetic energy of that molecule. So imagine this is my enzyme and here's my active site, my arm's here. I'm getting more and more kinetic energy, and that's actually starting to maybe start to break some of those bonds, and therefore my active site has changed shape. I'm no longer complementary to my substrate. That's not going to fit in there anymore. And I realise I look like an idiot, but hopefully it'll help you remember it. So that's what's going on there. pH, again, kind of similar. The idea here is that every enzyme will have its kind of optimum range for pH, where it works best. So an enzyme that's working in your stomach to help break down proteins, its optimum will be pH 2, so very low, acidic. But if you were to put it into alkali conditions, it would completely denature. Likewise, enzymes that work in alkali conditions would denature in acidic conditions. And the reason for that is that the hydrogen ions and the hydroxide ions, depending on whether it's acidic or alkali, will affect those bonds within the tertiary structure causing denaturation, that change in that active site shape, you're defining denaturation. Substrate concentration as well is an issue. If you've got a low concentration of substrate, so I put substrate there and then enzyme reaction there, you'll see at first substrate will very much limit the rate of reaction, so it will increase quite rapidly. And eventually what you would see is it would level off. So I'm adding more and more substrate, but why is it leveled off? Well, in this scenario, I've only got a fixed number of enzymes. So I kind of reach a point where all enzymes' active sites are occupied. They're kind of saturated at this point. A bit like, you know, when you go to the supermarket and you're trying to get to the checkouts, and if there's loads of free checkouts, people can get through really quickly. But after a while, everybody's going to be in a checkout and there's going to be queues forming behind them. And that kind of explains that leveling off. So here, before the leveling off, we would say that substrate is a limiting factor. Afterwards, enzyme concentration is a limiting factor. So that you can see how the substrate and the enzyme concentration affects the rate of reaction. Very much similar kind of shape. So that's a nice little overview there of our enzyme structure and function, factors that affect them. There is much more to look at, and I'm expecting you guys to look at your own notes, look in those textbooks, revision guides, so you're much more happy with them. But hopefully that's a nice little overview there for you on enzymes. One last thing to do, I'm sure you're probably ready to turn off this video now, but one last little area we've got to look at, I'll keep it brief, is about enzyme inhibitors. Right, okay, so the very last bit of our video then, I'm sure you're pretty bored now, but it's important we look at this last bit, and that is our enzyme inhibitors. So inhibitors are molecules that stop enzymes from functioning. They stop the active site forming enzyme substrate complexes in some way. And there's two basic ways they can do it. They can either be competitive inhibitors or they can be non-competitive. So our competitive inhibitors have a similar shape to that of the substrate. So always look out for that if you get an exam question or something like that. You'll notice that these molecules have a superficially similar shape to the substrate. So they can fit into that active site. And once they bind, they're out-competing any of the substrates. They're preventing any enzyme substrate complexes from forming. So obviously that's going to prevent that reaction from occurring, and it will slow the whole thing down, or even stop it entirely if your concentration of competitive inhibitors isn't high enough. Non-competitive inhibitors are very different. They do not have a similar shape to the active site of the enzyme. They have a shape that is complementary to another region of the enzyme, not its active site. The proper term is an allosteric site. And it binds to that region, and that causes the whole protein, the whole enzyme protein, to change shape. And more importantly, it changes the shape of the active site. So it's no longer able to form substrate complexes. Its substrate just can't fit anymore because the shape of the active site has changed shape. So that will stop the reaction as well, because we can't form enzyme substrate complexes either. The key difference here, and to tell the difference if you've got an unknown inhibitor and you've got an experiment to do, whatever, you're trying to figure out which one's which. Well, the easiest what I would do is add my inhibitor, add my enzyme, and then I would repeat the experiment with different substrate concentrations. And what you would find is, if you increase the substrate concentration, you would see, if it was a competitive inhibitor, the rate of inhibition would actually start to decrease as the substrate concentration increase, because effectively your substrate and your inhibitor are in competition with each other. And if there's more substrate molecules, they're much more likely to bind with the active site compared to the inhibitor. So you would see that happening. Whereas with non-competitive inhibitors, if that was the unknown inhibitor in your experiment, you would find that the substrate concentration shouldn't really have any effect on inhibition rate, because they're not in direct competition with each other. So that's two really nice looks at the old competitive and non-competitive. Now, inhibitors are found in your own cells. They're used to help control metabolic pathways, stop kind of runaway reactions from happening, so they're very, very important there. They can also be used in medicine. They can be used as drugs. If certain enzymes are overactive, for example, we can adjust those by giving you drugs that contain these inhibitors. And many poisons and toxins are also inhibitors as well. So if you think about snake venom, neurotoxins, a lot of those molecules in there are enzyme inhibitors, and they will stop a lot of your very important enzymes working, and they will prevent electrical impulses being generated inside your nervous system, which will effectively render you paralyzed and eventually, stop breathing. So it's pretty nasty, really, how inhibitors can be used. Very simple molecules, but they can be very, very powerful. So hopefully that's a nice little overview for you of the biological molecules topic. I know I can't get everything in there. I didn't get a chance to look at food tests, but I'm sure you can do that on your own. And I'll see you guys next week when we look at our nucleic acids and water in detail. So Dr. P signing off. You guys stay safe, stay indoors.